Membrane permeability. The plasma membrane, or the lipid bilayer of cells, is permeable to some substances, they can go through, and impermeable to other substances, they can't go through. And they are, to some substances, more readily to go through as other ones, and that they are selecting which come through is called selective permeability. But there are still ways to come through. Water can come through, and that is a bit strange, but first, why it's strange? Because things like uncharged and non-polar molecules, they can pass through. Carbon dioxide, oxygen, steroids. And so they're permeable to them. And charged things or polar molecules, like glucose or also ions, they cannot pass through. Water is charged. Why can water pass through? They're saying that it is probably, or maybe, or it is thought because of it, because all these glycolipids and phospholipids on the outside of the lipid bilayer, they're moving all around the whole time. It is not firm. A cell has not a firm part of the outside. These are just these phospholipids standing with the phosphates and the fatty acid tail, and they're all the time changing places, all the time. So one can go just in. And with the needle, for example, for fertilization or for cloning with dolicho sheep. So one can go just in, and the idea is now water, even though being polar, can just go in and through small gaps when they're changing again. That's the idea. There are some possibilities for other things to come through. There are the channels, and these channels are letting through only a specific substance, or also selective channels, letting them one substance through, only one specific one. A cell is oftentimes multiple different ones of these selective channels, so that through each of them can come a specific substance in. Or, for example, the transporters, they're taking something and like a snake, do they bring it in. And there is also the vesicular transport, with that also macromolecules, so macro, big molecules can come in, or other things. And that is not explained on that page here, it's later on explained. It has something to do with the ligand cord, that is the specific substance which can come to that receptor there. The ligand, some substance, comes into the receptor, stays from there. That would be just the receptor, and because it is there, it changes the surface of the cell. It changes the cell because the cell has now that ligand docked onto it. It changes the cell. But now the vesicular transport can happen. The clathrine-coated pit around the cells, they can start to go around and pinch them off, and now it's taken in. That would be vesicular transport, and that way also macromolecule or something could go in. And that is according to Gerard Tatoga and Sandra Grabowski's Principles of Anatomy and Physiology book on the page 64-65, but this is no substitute for medical knowledge and also for medical advice. Hi and welcome. I'm trying to learn cytology today, membrane permeability, and after we've done with it, we're going to do tiptoe yoga. And if you participate in the tiptoe yoga, then it's very at your own risk. And here's mentioned at the end of the section membrane permeability, macromolecules such as proteins are unable to pass through the plasma membrane except by vesicular transport. Discussed later in this chapter. And I already saw the next chapter is about transport across the plasma membrane, so membrane permeability. But we want to read now what I have written here about active transport. Secondary active transport. What is written about vesicular transport? A vesicle is a small spherical membranous sac formed by budding off from an existing membrane. The vesicular membrane is selectively permeable, and the vesicle contains a small quantity of fluid plus dissolved or suspended particles. So the vesicular membrane lets some things through, selectively permeable, and the vesicle itself contains a bit of fluid plus and dissolved also in which fluid plus particles which are dissolved or suspended. I don't really understand it. Vesicles transport substances from one structure to another within cells. Within the cell. Take in substances from extracellular fluid and release substances into extracellular fluid. Okay, we're taking things from the one side of a cell to the other side of a cell. We're taking things from the inside of a cell to the outside, and things from the outside in. Later in the chapter, we will discuss vesicular transport between structures within a cell. Once you have a better understanding of the anatomy of a cell. At this point, we will concentrate on the two main types of vesicular transport between a cell and its extracellular fluid. One is the endocytosis. In that, material moves into a cell in a vesicle form for the plasma membrane. Endocytosis, in which materials move into a cell in a vesicle formed by the plasma membrane, and endocytosis, in which materials move out of a cell by the fusion of vesicles with the plasma membrane. Both endocytosis and exocytosis require energy supplied by adenosine triphosphate protein. Okay, endocytosis is just into the cell and exocytosis out of the cell. Now, what is a vesicle? It is a small spherical membranous sac formed by budding off from an existing membrane. What is in it? Can there be something in it? Because okay. Endocytosis. In endocytosis, so something into the cell. The materials brought into the cell is surrounded by an area of a plasma membrane which buds off inside the cell to form a vesicle containing the ingested material. Here we consider three types of endocytosis: receptor-mediated endocytosis, pinocytosis, and phagocytosis. So the material brought into the cell is surrounded by an area of plasma membrane. Receptor-mediated endocytosis. This highly selective type of endocytosis enables a cell to take up particular ligands. A vesicle forms after a receptor protein in the plasma membrane recognizes and binds to specific ligand in the extracellular fluid. By receptor-mediated endocytosis, cells take up transferrin and iron-transporting protein in the blood, some vitamins, low-density lipoproteins LDLs, antibiotics, certain hormones, and specific other macromolecules or particles. Receptor-mediated endocytosis occurs as follows. We had in the previous chapter the transporting across the membrane, was it, or something transporting across the membrane. It was said about the receptors that there are specific substances called ligand or ligand, whatever, and that binds to there and it stays there. Now we're going one step further. So there's this receptor waiting for exactly a puzzle piece which exactly fits there. This ligand named like that docks onto it and it's not there. Now we're going one step further. We're doing the receptor-mediated endocytosis. So here now this thing goes around the thing. We're going to read it now. The receptor takes the ligand, the specific substance, goes around it and brings it into the cell. Now, number first, binding. So how it is actually happening. On the extracellular side of a plasma membrane, a ligand binds to a specific receptor to form a receptor-ligand complex. The receptor is standing there and waiting for its partner. And when the partner comes. The receptors are integral membrane proteins that are concentrated in specific regions of a plasma membrane called Catherine-coated pits, because the membrane is leaned on its cytoplasmic side by a peripheral protein called clefairin. Well, first thing, receptors are integral membrane proteins. That means they're reaching into the cell, into the membrane. And they are at a specific part of the cell altogether, of a plasma membrane. And this site is called Clefairin-coated pits, because the membrane is leaned on its cytoplasmic side by a peripheral protein called clefairin. Okay, at some place there's clefairin, there are all these clefairin-coated pits, and one of them are these integral membrane proteins, the ones who take it in, these receptors which are waiting for the ligand. What is it? Ligand or ligand? We're waiting for one thing and taking it in. Ligand. No, double A, ligand. The interaction between clefairin and the receptor-ligand complex causes the membrane to innervate. That means to fold inward. Oh. But I can't say that. Maybe I think that one is actually okay. Okay, the membrane is when making to invaginate. Okay, number 2, vesicle formation. The edges of the membrane around the clethrine-coated pit fuse and the protein of the membrane pinches off. Okay, the vesicle is not forming. The edges of the membrane around the clethrine-coated pit fuse. Clethrine-coated pit. The edges are fusing and the portion of the membrane pinches off. Oh, whatever, it goes around it and it takes it. It goes around it. Wait, I don't think I know what I'm doing. The ligand receptor is taking it, and then the clethrine-coated pit are taking it in. Okay, get it. The edges of the membrane around the clethrine-coated pit fuse, and a portion of the membrane pinches off. There's the ligand takes, and the other ones standing around it are all starting to connect to the one next to it, and that's where it takes it in. The resulting vesicle, known as a clethrine vesicle, contains the receptor ligand complexes. Yes, it's a clethrine-coated vesicle that contains the receptor ligand complexes. Yes, okay. So it's the other one where the receptor takes the ligand, and then all the other ones around starting to bind together and taking it in, because they're just going to go together or something. I can't really imagine it. If one goes to the one next to them, they're going closer together, obviously. And if the next one goes closer together, they're also going together. And when they're forming around it, uncoating. Almost immediately after it is formed, the clethrine-coated vesicle loses its clethrine coat to become an uncoated vesicle. Okay, they are shedding the clethrine. Okay. So what the thing which has taken them in, that is taken away, and it's just a vesicle. 4. Fusion with early endosome. Several uncoated vesicles fuse with an early endosome, a vesicle that serves as a sorting compartment. Where does that come from? Okay, whatever. There's someone waiting for them on the other side. An endosome. And they are sorting them all together. Within an early endosome, lignans separate from the receptors. So the lignans was just going in the receptor, and then they're splitting off again. Ultimately, the contents of an early endosome are either recycled to the plasma membrane or transported to a lyosomal organelle where degeneration occurs. Was the endosome explained? Whatever. There's an endosome that takes them, the lignans are going away, then the endosome takes them, and the endosome takes multiple of them, and the contents of the early endosome are either recycled or degenerated. 5. Recycling of receptors. Many receptors within the early endosome enter a carrier vesicle which buds from the early endosome and returns the receptors to the plasma membrane. Clethrine also is returned to the inner surface of the plasma membrane. Okay, they're both recycled. The clethrine, which was taking it in, was set in, is taken from another carrier vesicle back to the outside, to the plasma membrane. And the receptors are also. 6. Degeneration in lyosomes. Ligands and the receptors destined for degeneration in lyosome are transported from the early endosome by one or more carrier vesicles to a late endosome. Okay, they're transported further on. The late endosome forms other carrier vesicles that fuse with lyosoma. Within lyosoma are digestive enzymes that break down a variety of large molecules such as proteins, polysaccharides, lipids, and nucleic acids. Okay, we're working on it, and we have received it. Transcytosis. In some cases, ligands and receptors undergo transcytosis, a process whereby they are transported across the cell inside carrier vesicles that undergo exocytosis on the opposite side. As the carrier vesicle fuses with the plasma membrane, the vesicle contents are released into the extracellular fluid. Transcytosis occurs most often across the endothelial cells that line blood vessels and the means for materials to move between blood vessels and interstitial fluid. Is it just an accident? Is it just an accident? So what we're describing here was the receptor is taking that specific ligand to it. then it stays there. The caffeine or whatever around them are starting them all bind together and they're bringing it in. When it goes happily through the cell, and when the caffeine at the outside have brought it in, they're going away, are transported maybe again back to the outside, doing the job again. And when the cell goes on, and maybe it comes to the other side of the cell, and when it is released to the other side of the cell, and it goes into the outside. It's not released inside the cell, but to the outside, and that way something was transported through it, or at least or also outside. Whatever. The question is it just an accident? Or a planned-in accident? No, we don't have time left. We're going to come back in the next part, and we're actually going to read on the rest of this around the page. In the same way, and when we're going to go back, and then we're making a video about the brain permeability, so that we at least have read what is vesicular transport. We're not going to really explain it, and we're going to do the vesicular transport later. If you're here for Cytology, you can go directly to the next part, we're going to read on. If you stay here and participate in the short tiptoe yoga, then it's very honest. The feet are standing together, believe in the arms up. Come up onto tiptoes. Out and down, and come always to the big toes of the weight. In and up. Out and down. In and up. Out, up to the side. In and up. Out and to the side. In and up. Out and down. And the arms up. Make an inch space between your feet. Out and to the side. In and up. And out, sink down. In and up. Out, up to the other side. In and up. And down. In and up. Out and down. And one toe points to the outside. The other one in front. Breathe in, bend the front leg. No, breathe in. Out, bend the front leg. Come up on the tiptoes. Breathe in, reach your front leg. Out and back. In, straighten the front leg. Out and down. Down. Change the legs. Breathe in. Out, bend the front leg. Breathe in, reach your front. Come up to the tiptoes. Out and back. Breathe in, straighten the front leg. Out and down. Back. Down. Feet together. Breathe in the arms up. Out and down. Bend the right leg and stretch the left one. In, change. The right one. Change middle. Down. Open the feet to the sides, but keep down the reach again in. And out one leg. Breathe in. Out to the other leg. Breathe in. Out middle and close the feet. Breathe in. And outward slowly with the round peg up, leg at the top, backwards, shoulder down. In the arms up. And down. That was the tiptoe yoga. I thank you very much. Until next time.